So the title of this talk, as you can see, is it's about the development of velocity selective ASL fingerprinting. And I'm particularly excited to give this talk here in this forum because the Hopkins groups from Han-Sang Liu and Chin Chin have been in the foreground and the cutting edge of the development of both velocity selective ASL and fingerprinting. So what I'm hoping to do is to share my experience with these techniques and maybe to, instigate a little bit of back and forth and maybe a little brainstorming and have some good discussions on it. And maybe something will come out of this that'll be greater than the sum of its parts. So I guess I'll start out by trying to just kind of give a little background of why we're doing what we're doing, and mostly it's that blood flow is a very powerful indicator of tissue health. That's a really obvious statement when you're talking about vascular diseases like strokes or moya or some sort of arteriovascular malformation. It's becoming increasingly clear that the vasculature and the ability to deliver nutrients and clear out waste is really important in neurodegenerative disorders, and there maybe even in a causal way. There's cancer, of course, where blood flow is altered because it follows metabolism so well. But also a lot of body imaging is happening around the kidney and liver disease, where blood flow plays an important role. But more important and dear to my heart is that blood flow is also an indicator of activity because metabolism demands blood flow. And when there's more activity, there's more metabolism and the blood flow goes up. And this is what allows us to do fMRI. And a very interesting thing is that that functional MRI done with blood flow related techniques like VASO and so on actually allows you to do, it's much more specific to the activity than the bold effect is. So that's also a very exciting motivator. And needless to say that, you can also look at the activity of other organs when you do challenges and so on. So there's like a whole wide range of applications to these techniques that are just scratching the surface right now. So another part of it is sort of philosophical, and it's that doing quantitative blood flow imaging really adds a lot more rigor and reproducibility to a radiological examination. Just a qualitative blood flow map is already helpful to make diagnoses. But when you want to study populations and so on, you want to be able to reduce the variance of your measurement, or be able to achieve some standards by putting numbers on those blood flow images. Very good. So I'm just looking at the hooray for quantitative imaging. So our objective here for this talk and for the work that I've been doing for a long time is to develop new blood flow imaging methods. And we want them to be non-invasive. We don't want to have to inject anything into anyone. We don't want to use radiation. And of course, we want what everybody wants, high resolution and these isotropic voxels. We want to be quantitative and we want to get a lot of nuance. So we want to be able to get lots of parameters in a single scan or a single scanning session. So of course, especially with this audience, you're all thinking arterial spin labeling. And yes, it's a great alternative to doing tracer injections and shooting gadolinium into people. In a nutshell, what you're doing is you are doing a perturbation of the magnetization in the arteries, and then you observe the magnetization in the tissue and the capillaries as that perturbation that you create in the arteries gets into the tissue and the capillaries. And you can wash it in and out. And this stuff's been around forever, since 92 and so on. But it's a hard thing to accomplish, and it's taking a long time to reach maturity and to get to a point where it's actually usable. And we're still developing and improving it. So let me tell you the basic anatomy of an arterial spin labeling pulse sequence is that there's going to be a labeling period where you're doing that perturbation of the arterial blood. And then there's a lot of different options that you can have that we're going to talk about. And then after a little bit of a delay, those perturbed spins are going to get into the tissue. And then you can have different kinds of preparations, like getting rid of the arteries or adding T2 weighting if you're interested in oxygenation. And then there's going to be a readout component of this. And typically we try to do this quickly before the label washes away. And for that, there's been all kinds of trajectories that have been tried out. The dominant ones are the 3D Cartesian ones, but there's also, stack of spirals, which is my personal favorite. There's radial and Kuchball techniques and so on. So there's a lot going on here. Okay, so the classic recipe for the quantification part of this, now that we have images, is that, well, you collect a pair of images, one with a label, one without the label, a control image, and then you subtract them. And you also need a spin density image for reference, so that you know what fraction of the spin density has been altered by the labeling process. We also need to know the T1 of the tissue, so you'll need some kind of inversion recovery experiment, or you can just sort of assume values with whatever error that entails. You're going to have to clean up and co-register all the images into the same space. And then all of this is going to go into a formula that's going to give you a blood flow map. And this formula is based on a kinetic map. So we can get more nuance. We can get more things like, the bolus arrival time. How long does it take for that bolus to even get to the tissue? And that tells you about the health of the larger vasculature. You can quantify the blood volume, what fraction of a voxel is in the arteries and the veins. And each time you do this, you have to repeat the experiment and vary one parameter at a time. So it can get pretty slow. And you can do this for multiple other parameters. You can do this for measuring oxygenation or velocity. People have looked at the permeability of the vessel walls. So there's a ton of information in this arterial spin labeling technique. But the caveat is that you have to repeat the experiment multiple times and then put all that information together. So it gets very long, and the errors and the uncertainty, the noise that it's in each one of these techniques keeps accumulating. So, there's some real challenges here. One of them is that it's a low SNR technique in the first place, because it's a very small fraction of the voxel that's actually contributing to the signal due to what the perfusion rates are. Also, this label is decaying during the transit between the time you create this perturbation in the neck, in the carotids, and the time it arrives at the tissue. So there's a lot of T1 decay there. We're also subtracting images, and this sort of thing always amplifies your noise and the artifacts, particularly movement. Movement can be a real killer. And since you have to, as I was alluding earlier, so you have to have all these additional measurements, there's a lot of error propagation that happens. And then you end up with a lot of uncertainty and a lot of extra scan time. Okay, so with that, we kind of look back at our goal of achieving this new blood flow imaging method, and we have some really nice new techniques in the horizon. Well, it's not really the horizon anymore, they're here. And one of these is MR fingerprinting, and then there's a lot of really exciting optimization techniques. And then, of course, there's velocity selective labeling pulses, and neural networks are showing up everywhere. They're taking over the world. And we're finding out also new ways to read out the data and to do more with less data. So we're trying to bring this in, and that's sort of the goal of this talk, is to integrate a lot of these things. So we're going to start out by talking about ASL fingerprinting. And mostly I'm going to try to highlight this last paper from my student Anish who did a fantastic job with it. So MR fingerprinting without ASL kind of showed up in the scene in 2013 in this Nature paper. And the general idea, the strategy of it is that we're trying to quantify MR parameters, things like T1, T2, and spin density. And before fingerprinting, we would have done an experiment trying to hit a steady state, and repeating it over and over, but each time would be varying one parameter at a time. Like, for example, the TR, or for example, there would be an inversion pulse, and then you'd vary the TI, right? And you'd have to repeat this, and then you'd fit a curve to a very simple model, and that would give you T1 at every voxel. But the fingerprint actually tries to work backwards. What the general philosophy is, we have really good forward models that use the block equations that can predict the signal for any combination of parameters, T1, T2, and 0, blah, blah, blah. And if we come up with a crazy pulse sequence where there's no fixed TR and no fixed flip angle, but rather we do variations. So for example, in this figure from their paper, we do a time series of images, so it's 500 images, and the flip angle is just, doing this crazy oscillation, and the TR is just completely random. So this is going to generate a signal like it's up here in the right upper corner. And what's really interesting is that it's going to be a unique signature for a specific combination of parameters. So instead of trying to come up with a complicated search algorithm for the right combinations of T1s and T2s, you pre-compute a dictionary of combinations of underlying parameters, and then you try to find which one is the best match. It doesn't have to be a perfect match, it just has to be the best match. And indeed, what happens here is that this red one here that they have is the signal in the dictionary, whereas the blue one is what you actually observed. And it's a terrible fit, but it is the best fit, or the most correlated one. And the images themselves, especially with a pulse sequence like that, and with this kind of level of undersampling, they look terrible, but it's still the best match. And from just by doing this, you can actually get very decent T1 and T2 maps, as well as spin density maps, and even off resonance, just by calculating this dictionary. So it's a very powerful technique and very promising. And there's been a lot of development, a lot of validation. And the key concept is that you don't have to work with a steady state like we normally do in MRI land, or we used to. But we just have to achieve some kind of dynamic state. And then instead of fitting the data, we're just going to match a database of previously simulated signals. So if we try to look at the ASL case, we can also have a block simulation that will include perfusion, bolus arrival time, arterial blood volume, like all these other parameters, and we should be able to estimate them all at once using this sort of fingerprinting technique. So this may very well address a lot of these concerns and challenges to ASL. It seems to be robust to a lot of artifacts. That's one of the things that they demonstrated in their fingerprinting papers, like undersampling and movement and off resonance. And it might solve all this issue of having to put all these different parameters together, all these co-registration errors, and all the multiple parameters that you have to fit from a single scan. So you can just put the patient in, get one time series, and be done with it. And then just kind of put it through the machinery, and that will give you these nice quantitative maps. So this is indeed very appealing. Okay, so let's talk about building a dictionary and the signal model. So if you kind of follow the ASL canons and recommendations lately, you're probably using this Buxton model to fit the ASL signal. And essentially, it's a model of the tracer kinetics of the difference in magnetization in a steady state. So for simulating the dynamics for fingerprinting, we're going to have to actually go back to the original block equation models that they used in the very first papers of ASL, the ASL technique. And it's essentially just the block equation with your T1 relaxation, but you include, flow terms for inflow and for outflow. And then, of course, you have to account for the fact that there's going to be a lot of magnetization transfer that arises from the labeling pulses. Okay. Any questions so far? Also, please do not be afraid to interrupt me if you want. I'm very happy to just make this more interactive. It also lets me know that you guys are awake. Anyway, so let's with that, let's see an example of. So here we're modeling a PCASL sequence, sort of the standard. And on the top panel, I'm showing the just kind of in broad strokes the important parts of the sequence where I'm applying control pulses. Then there's a readout on these little red spikes, and then there's a label, and then another readout, and so on. And when you read out, you perturb the magnetization in the arteries, but that washes away, and you perturb the magnetization in the tissue. So you tip down, and then that has to do it's T1 relaxation. And during the control, you notice that there's an magnetization transfer effects, which is sort of essentially kind of killing off a little bit of your magnetization. So that's something we account for. And then when you apply the labeling pulse, what it's doing is it's taking the arterial magnetization and it's bringing it down during this bolus, during the input. And you notice that there's a delay from the bolus arrival time. So this kind of framework can really catch that. Over time, we do this, label, control. And here where we do the tip down, that's where we obtain our data, and then we can catch the different time points throughout the evolution. And this is what we end up observing. This is what's going to make up our fingerprint. So we tried this out. We've been kind of working on this for a long time. First, by coming up with a pulse sequence where the labeling durations and the post-labeling delays were variable, and we just kind of came up with some combinations. We used this sort of similar models. And doing this sort of thing, we were able to get some nice T1 maps, as well as perfusion maps or CBF, the time to peak of the tissue and the arterial time to peak, as well as the B1 map, what flip angle we were actually getting, and the blood volume. So that was really nice for a single slice. At the same time, Han-Sang Lu's group here at Hopkins was also working on it, and they came up with actually a better way of doing it, which was to do away with the post-labeling delay altogether. And essentially, they considered they used the control pulses as post-labeling delays that were variable. So I thought that was a very, very clever idea, and it worked really well, and they got some spectacular results, and they've been working on this increasingly and improving upon it, and getting some really nice results. Okay, so there are some problems with this dictionary matching approach. That one of them is that these dictionaries get really large, and the more parameters you want to estimate, then that makes a dictionary grow even longer exponentially. So in order to make it manageable, your dictionaries end up being a little bit coarse, meaning like in terms of the sampling rate of that dictionary. And then it turns out that there's a lot of correlation among the entries when you're doing spin labeling. The spin labeling effect is very subtle, so the T1 dominates, and the signals for two different flows end up being fairly correlated with each other. So there's more sensitivity to some parameters but others. So maybe we can do better. So we kind of go back to the drawing board, and the things that we come up with is that, well, first of all, we should try to optimize the timing schedule. We need to, rather than just kind of guessing and doing this cognitive optimization, we need to be able to do something that's more formal and more rigorous. And that will allow us to increase the sensitivity to the parameters we want, and thus reduce the variance of those parameter estimates. And then let's just ditch dictionary matching and see if we can use neural network regression, because it's not going to have these discretization limits, and they're very fast. Once you've got them trained, they give you very fast response. So the next stuff that I'm going to tell you about is from this paper that we published on the subject in this reference here. Okay, so let's talk a little bit about the optimization first. So our goal is to design a timing schedule, essentially a series of labeling durations that will go for our time series, right? And we want to maximize the sensitivity of the signal to the parameters of interest. So, you know, changes in blood flow, for example, will give us a very different signature than will give us a very, very big change in the signature. Okay, and for that, we have this tool from statistics called the Kramer-Rao lower bound, which turns out to be very useful for this sort of thing. And it is defined as the inverse of the Fisher information matrix. And let me just kind of talk a little bit about that. If I can get my things to advance. Okay, hello. Okay, so here's the definition of the Fisher information matrix. And what it is a matrix that you construct by taking partial derivatives of the signal model with respect to each of the parameters of interest. So each one of the entries is essentially giving you the rate of change of the signal as a function of the rate of change of the specific parameters, right? And then in our case, we make a distinction between the parameters we want to estimate, blood flow, the blood volume, and so on, and the parameters that have to do with the pulse sequence. So we're really interested in the partial derivative relative to the parameters that we want to estimate, these thetas, guys, right there. And by calculating the inverse of this matrix, you can get a measure of the minimum variance that you can possibly achieve by estimating these parameters, giving this amount of noise in the system. Okay, any questions there? So that's our measure of goodness, our cost function, right? So, but the actual optimization is we want to search for the acquisition parameters. In other words, our schedule of labeling delays that's going to minimize this Kramer-Rao lower bound, or this minimal standard deviation that we can achieve during the estimation. So we're trying to minimize the trace of that Fisher information, of the inverse of that Fisher information matrix. And then, as I was alluding to earlier, some of the parameters have more effect than others. So if we leave this alone, we're pretty much going to come up with a schedule that's going to give us really, really good T1 estimates because they dominate. They're the thing that we're most sensitive to. So we have to come up with some sort of weighting matrix or weighting vector that's going to emphasize the blood flow parameters, which are harder to estimate than the other parameters. And we just kind of build that into the cost function, into the optimization process. Okay, so now we have a nice cost function and a minimization problem. Now let's talk a little bit about the search. So the way we searched for it is we constructed, say, well, we're going to get 700 images, and we're going to select these 5 control points, and we're going to just look for different values at these control points of the labeling durations, and essentially interpolate between them. And all put together, it gives you a nice function that describes the schedule of labeling durations throughout the time series. And another thing we were trying to do is we were trying to make this thing be 10 minutes long, no matter what. So in doing so, we would always rescale it so that it would always add up to 10 minutes. Okay, so the actual search was actually not very sophisticated. It was a grid search. It was, I guess, 5 by 10 combinations of schedules that we could search for, and we were just trying to find which one was giving us the best Kramer-Rao bound. And this is what came up. What came up is later. So once we had that optimized, then the next step was to design a neural network that would be able to solve the inverse problem, essentially a regression. And at first we tried making a neural network that would spit out all the parameters at once. But then that became pretty unwieldy because, again, we had to weigh them differently. And then we learned that it was just easier to make one neural network for each individual parameter and just kind of train the network, train each one of these networks to solve for one parameter at a time. Okay. And so the way we did it is we simulated 6 million time courses with the variation of blood flow parameters, and we trained the network. We trained the 6 networks to give us the parameters we wanted. Okay, so this worked pretty well, at least numerically, doing simulations. So we kind of made this numerical phantom based on template image, and we just kind of assigned flow values and blood volume values, bolus arrival times, magnetization transfers, T1s, and flip angles. So we just kind of created these synthetic maps, and then using them, we generated signals, we added noise, and by putting them through a network, we were able to get back the original maps pretty well with some bias, but not too much. The same plots here with plotting the truth versus the predictions, they were pretty faithful. They were very good about getting T1s and flip angles and magnetization transfer rates. But with the blood flow parameters, you see that there's a little bit more scatter. And with the bolus arrival times, as the bolus arrival time gets bigger, so does the error. The error gets really big, and it makes sense because at very long bolus arrival times, you start losing your label, and there's just not much information left there. I see a question on the chat: solving for one parameter at a time in the case you can estimate the non-CBF parameters first, and then use their best estimate values for estimating CBF. Aha, that's a great question. So this came up. We kind of thought about this quite a bit. So one approach would be to, let's say, for example, solve for T1 first, and then now that we know T1, we could just train a network that has been constrained to one specific T1, and reduce the search space and give us a better answer, a more defined space. So that would work. That does work. And the problem with it is that the training is very slow. And then we're talking about having to train the network, having to train a different network for each voxel, essentially, as the T1 varies and so on. But it's certainly a good idea. Okay, so anyway, so this kind of worked in vivo, in silico, and we tried it out in vivo. And we try to do a validation of sorts by comparing it to a multi-PLD method, where we could fit either a 1-compartment model or a 2-compartment model. So these are the maps that we obtained with our method, with the flow, CBV, and so on, and they correlated pretty well with the multi-PLD stuff. And then we wanted to answer the questions like, well, was it worth the optimization? So we did kind of a randomized post-labeling duration scan, and then a post-labeling duration schedule that was decreasing monotonically. And the one that was optimized was clearly performing better and correlating much better, especially with the 2-compartment multi-PLD model. So that was very good news. But it's still not great. This was a single slice. It was a very long scan time. The validation, we could validate, as you saw, most of the parameters, but not all of them. And the model is very complex, and there's some things in there that, that are nice to see, like the magnetization transfer, but that wasn't really what we were after. And also, we had these arterial transit time, and then there was sort of a tissue or capillary transit time, and maybe we don't need both of them, so maybe we have too much complexity. So we're kind of thinking, well, can we do better? And I think we should be able to do better. So this is where velocity selective ASL comes in. And and at least this quote unquote chapter, it's going to be about like, okay, how to do velocity selective ASL, how to choose a good velocity selective ASL pulse, and then I'll move on to bringing this into the MRF framework. Okay, so in a collaboration with Jia Guo in Riverside, we try to answer this question. So velocity selective labeling, essentially what we're doing is we're labeling the arterial spins, but instead of rather labeling depending on the location, we're gonna try to label them depending on their velocity, whether they're stationary or moving or fast or slow. So there's some real advantages to this. One of them is that it requires a lot less time for the tissue to get to for the label to get into the tissue. So this means less relaxation, less loss of the label. And these pulses are also nice because they don't exhibit any noticeable amount of magnetization transfer, so we can drop that from the models. There are some challenges in that these pulses, they're not perfect. They're sensitive to B0 and B1 imperfections, and you don't get the kind of efficiency as you do with the PCASL because of T2 effects. And I'll talk a little bit more about that in a second. So just to recap, something that you probably already know, when you're doing a continuous ASL experiment, you're applying a labeling pulse over here, the blood flows in, and then you collect your image. When you're doing a velocity selective experiment, you apply a pulse that affects the entire region, but what's really giving you the contrast is what's in the arteries. So the label was already here in the right location, it was just in the arteries, and then you kind of watch it getting to the tissue, and then you collect your image. So the kinetics that you can get from this, if you look at just the arterial input function up here, in these velocity selective pulses, you don't have a delay, right? So and it's very quickly decaying with T1 as the back end of the bolus comes in. Whereas in PCASL, you had this long delay, but you had the nice feature that the input was constant as long as you had, the label on that translated into the width of your bolus. When you take this arterial input function and look at what's getting down to the capillaries and the tissue, you get these really nice curves. So you can see that there's some real advantages, particularly to the velocity selective inversion pulses. So these velocity selective pulses, they were first introduced in '98 by Norris and Schwarzbauer, who started kind of developing a little bit of the framework. And then then they really got into the foreground when Eric Wong's group introduced them for ASL with a kind of a new design where he was doing dual refocus hyperbolic sequence velocity selective saturation. It's a bit of a mouthful, but it's essentially a couple of a 90-degree pulse, two 180s, and a -90 that could tip the magnetization down, kill it if it's moving, and if it's not moving, bring it back up. And then later on, his student Jia Guo, but also James Meakin and I believe it was in Oxford with Peter Jezzard, came up. They got together and they came up with a better version of them using these BER8 pulses, these adiabatic pulses that could essentially achieve the same effect with a lot less artifact. And with these, you're able to saturate the spins that are in the arteries while not doing anything or not doing too much to the stationary spins. But then what was really cool was a paper from your group, from Chin's group on Peter Van Zyl on Fourier transform velocity selective pulses. And they essentially figured out that you could excite moving spins following a velocity k-space pattern, so that if you, for example, played this pulse that broken into chunks, so this would be a rect pulse. These little skinny guys are pieces of a rect pulse, just a nice square wave, and you could stick in some refocusing pulses in between. You could actually encode velocity by having a train of gradient pairs. And and this could give you a velocity pattern. So the paper came in 2014, but they presented the stuff in ISMRM in Milan, I think. And I was really blown away. I thought this stuff was brilliant. So I said, whoa, can we try an adiabatic pulse and do the same thing? So you can take a hyperbolic secant with, you know, the characteristic phase pattern of it. You can chunk it up and sticking a velocity encoding pattern, and you can get really nice velocity selective adiabatic pulses. I see a question: are the velocity selective pulses directional? Yes, they are, and all the velocity encoding happens in the direction of that gradient. Typically, this is done along the z-axis, and one thing that's kind of a little bit surprising is that it is in the human brain, at least. It turns out it ends up making very little difference whether you pick the x, y, or the z-axis for your gradient selectivity, and the reason is because there's always kind of a distribution of velocities, and there's a lot of tortuosity, and it's like you always end up hitting them, you know, no matter what axis you pick. There's some subtle differences, but it turns out to be really small. It was surprising, but surprising but true. But yes, if you have tubes of water and you move them perpendicular to the direction of the gradient, you will not get this effect. But in the brain, you do. In the brain, there's enough movement around that you can get, that it doesn't end up mattering so much which gradient you select. Okay. So here's like a simulated velocity profile for one of these pulses. So this is the rectangular based velocity selective inversion pulses that Chin came up with, and you can see it's essentially a sink shape. It's the Fourier transform of the rect that they were using. And what you see is that when you apply a control pulse in which you're not using any velocity encoding gradients, everything gets inverted. But when you apply these velocity encoding gradients, you only select either the zero velocity, inverted of zero velocity, or you know, you get these aliasing bands, because it works just like phase encoding. You know, there's going to be aliasing patterns and periodicity. So the label that you achieve is really the difference between these two guys. This area is essentially your bolus right there. Okay. So after a while, a lot of pulses are evolving, and there's been some experimentation, and then we kind of come to like, well, which one's going to work better, the original velocity selective pulses or are these, because they're not very perfect. So the inversion pulses at a first approximation, they're gonna definitely do better because you get twice the label. But it turns out that the saturation pulses, they have kind of a nicer velocity profile, and they're a lot less sensitive to imperfections. So it ends up being kind of a difficult question to answer, and we try to answer it. Gia and I had a lot of discussion on this, and we tried to do kind of a formal comparison of pulses. So on the left corner were these velocity selective saturation pulses, and on the right corner, and the real hogs, they're actually taking a lot more than the right corner. We have all these velocity selective inversion pulses, Fourier transform velocity selective inversion pulses, and they follow different velocity selective patterns. But we try to match the width of the central band, which is where most of the action is. Okay. The thing that's really important to consider, and the reason these pulses work at all, is that if you look at the velocity pattern, it's oscillating like a sinusoid, and this is not very useful. But if you take into account the fact that flow is generally laminar in the vessels, so there's a whole distribution of velocities, and once you integrate that over a laminar velocity profile, you get this very nice sync pattern on the velocity selective saturations, where there's going to be a cutoff. We define a cutoff velocity as where the place where the saturation happens at the zero crossing. And the same applies to the Fourier transform pulses, and you get this other curve like that. Okay, so then we started with simulating what different kinds of imperfections can do to these pulses. So in here, in this beautiful color map, we're looking at that velocity profile, but we're color coding it. So blue means inverted and red means uninverted and completely relaxed. So, and on this axis, we were changing the B1 scale, meaning like up here, we had 70% of what we thought we were putting out. And over here, we had 130% of what we should have been putting out. So, and then we did the same thing with off resonance, just adding different amounts of off resonance. And then we also added eddy current effects with different time constants. We were looking at, what the effects were over different velocities. Okay, any questions on this? There's a lot of information in these plots, and they're kind of a little bit difficult to parse. The ideal pulse would just give us nice straight blue lines at the target velocities, and everything else should stay red. Okay, so here's the big super colorful plot where we looked at all the different pulses, our velocity selective saturation ones, and then our Fourier transform VSI pulses. And we see that the sync-based VSI pulse was really performing the best, like next to no eddy currents and very robust to off resonance and to the B1 scaling problem. So that was very nice, very convincing stuff. So then we, by examining these a little more carefully, we examined the features of these different pulses, their charming characteristics. And one of them is that you don't get perfect labeling efficiency. You never take the spins all the way to -1. And the reason for that is that it's kind of a slow process, and there's always a component in the XY plane, and whatever components in the XY plane, it's going to be relaxing with T2. So you have a lot of loss to T2. So they have kind of an effective echo time and T2 loss that gives you this much efficiency loss. And then when we integrate over these different imperfections in those plots, we see that some of them are more sensitive to the imperfections than others. Additionally, one thing that you get is you get a lot of diffusion weighting added from the pulses because you're doing this train of RF pulses with different refocus gradients, and that's going to have a significant B value that's going to affect your signal. Okay, so we look at this in vivo and looked at, I believe, 9 subjects, and we compared it with PCASL. So PCASL serves as a reference, and we looked at how well our CBF maps, after we put them through the quantitation steps, how well they correlated with the PCASL quantitative maps. So the ones that stick out were like the VSI sync. So this is the one that was actually performing the best in terms of being robust, but also the a dual velocity selective saturation pulse. And essentially, this is just the velocity selective saturation, but only if we apply 2 of them. And that gives you, that increases the amount of label that you can create. And this is, and Gia has a very nice paper developing this technique with the dual velocity selective saturation pulses. So these 2 seem to be the ones that had the most correlation with PCASL. One of them seems to overestimate the flow a little bit, and the other one seems to underestimate the flow a little bit, telling us that some of our assumptions somewhere, there's a little systematic scaling factor somewhere. If we kind of dissect that performance a little bit more, and we look at just the amount of signal that you get, one of the things that jumps at you is that the original rect pulses, well, they had some sensitivity to off resonance. that will give you these hot spots that are not really flow. So they show up as high SNR, but they're kind of artifactual. So we were not too pleased with that. In terms of the temporal SNR, these sinks did really well. They seem to be pretty stable. And so here's averaged over the group. And on average, the performance was not dramatically different between all these pulses. And that was much to our surprise. We each had our favorites. And we were kind of expecting them to come out head and shoulders above the rest, but it's not a head and shoulders difference. And then, some correlated better with the original PCASL and others. So the conclusions that we drew from these messy information was that the rect VSI pulses had the highest SNR, but it had artifacts in it. So that was not the favorite. The two favorite ones were the sync VSI and the dual velocity selective saturation. They seem to be the ones that agreed most with our quote unquote gold standard, the PCASL. But one of them seemed to underestimate the flow a little bit, and the one seemed to overestimate the flow a little bit. Not too bad. And one feature that we noticed is that the velocity selective inversion pulses, because they're inverting the magnetization in the stationary tissue, they're contributing to the background suppression, and that's an advantage that they have. And that contributes to the increase in SNR, because they're suppressing that background that contributes to the noise. Okay, so very good. Now we've studied velocity selective pulses. We kind of figure out a good one. We're gonna use the Fourier transform sync-based velocity selective. And now let's see if we can put it together with the fingerprinting scheme. So on that, we kind of made some progress, and we presented it at ISMRM, but it's still a work in progress. So we used, again, another MRF sequence that was using these velocity selective pulses. And we had to re-optimize it, and we used another model. And I'm looking at the time, realizing that I'm running a little bit long, so I'm gonna speed it up. But this is essentially the same idea as before, with a similar model, but adapted to this. Only this time we had to adapt the labeling efficiencies to reflect what these FTVSIs pulses are doing. After the optimization, we came up with this labeling scheme, and again constrained it to 10 minutes, and we fixed the prior interval to the label to 2 seconds. We trained the algorithm with another 6 million time courses, where we varied 6 different parameters, this time the different efficiencies on the arteries, the tissue, also the efficiency of the saturation pulses. And we were able to get some decent results on perfusion, bolus arrival time, and T1. It's worth noting that the bolus arrival time doesn't mean the same thing as PCASL. Here, this bolus arrival time is that time to go from being to the time it takes to slow down from being in the arteries to getting into the capillaries. And it follows kind of a different pattern. And it's something that's going to be kind of hard to validate. We can improve on this. I think we can do better. I think the signal model could kind of use some more scrutiny. We need to include blood volume into the model. And we also need to focus now on the 3D readout, and that's kind of what we've been kind of putting all our effort in. And then we'll have to re-optimize the labeling schedule and then validate it against the gold standard. So I think we can do better, and we're working on it to see if we can just kind of redesign the whole thing and kind of give you a nice robust technique. There is a nice side effect to this PCASL fingerprinting, and it turns out that it's great for tissue classification. If you track the signals of the different gray matters, you see how they look very different. So this should make it very easy for us to identify what's CSF, what's gray matter, and what's white matter. And we were able to do that mostly because we're interested in doing partial volume corrections, which are becoming much more increasingly important and playing a role in ASL. So usually you get a T1 map and you put it through whatever morphometry package you're using, and it spits out this correlation maps. I'm sorry, these tissue segmentation maps. Okay, but the problem is that then you have to co-register the images, and sometimes they require these templates, and they may not necessarily match your subject's anatomy very well, and you get a whole bunch of errors from it. So I think it's great that we would be able to just do the segmentation from the MRFs themselves. So we can do this using a self-organizing map that essentially use this reference to come up with the algorithm. And all you're doing is just starting with some exemplar pulses, time courses, and then adding the different voxels into it until the exemplars converge to a canonical time course. And then the second stage is now that we have these canonical pulses, we just do a linear model and try to see how much of the voxel signal is made up by each one of these canonical courses. So to illustrate it, these are the components that emerge from the self-organizing map. And then when you do the linear regression, you get these nice maps of the fraction of the signal that is accounted for by each one of these components. Okay, so this worked pretty well. We've done that a few times, and we're getting some very nice tissue type classifications that seem to, at least on a first look, they look like what you expect CSF, gray matter, and white matter maps to look like. And then we've eliminated the need for additional images. We've eliminated the need to go through this normalization process and through this co-registration process, eliminating those errors. And so on. So of course, we still need to validate it. We're still kind of in progress, and we need to actually take these partial volume maps and integrate them into the excuse me, into our workflow of trying to get the parameter maps. Okay, so where are we going next? Well, we gotta finish all the work that I've been telling you that is half done, for one thing. But where we want to go is using compressed sensing to use to undersample case space in different bits at different times, and then combining this partial case space information with our different multiple coils, and letting a neural network do this combination and doing these interpolations, and hopefully by some black box magic, we'll be able to spit out these perfusion maps, T1 maps, CBV maps, and so on and so forth. And that's where we're going to go. So with that, I realize that I'm just over time. I'd like to thank you for your attention. I'd like to thank my collaborators, both at Michigan and also at Riverside, Giaguo, and also Mark Griswold and Vikas Bulani. Vikas is now back at U of M with us, but they were the fingerprinting guys. And also former students and my two current students, David Fry and Tao Hong. Actually, Tao Hong's a postdoc, who are doing fantastic work. And thank you for your attention.